In Chapter 6, we get to talk about two key aspects of metabolism, energy and enzymes. We begin with energy. So we're aware of the fact that there are two forms of energy. Kinetic energy, which is the energy of motion, which is a form of mechanical energy, and we see kinetic energy displayed on our slide here in the bottom picture as this ball rolls down the hill. The other type of energy we have is potential energy, which is a form of stored energy. Now, it's easy to think about potential energy in the form of this ball sitting at the top of the hill because it could be pushed and rolled down the hill. But what we need to become accustomed to thinking about is potential energy in the form of chemical energy, which is going to be the form of energy that we deal with here in the world of biology. To understand these two forms of energy and how they interact with biological systems, we need to understand and be familiar with the first and second laws of thermodynamics. The first law is the law of conservation of energy, which simply states that energy cannot be created or destroyed, but rather it can change forms from one form to another, and we see this illustrated very powerfully as we look at the natural world. For instance, take a look at this picture from our text. We see the sun, and as we pointed out in lecture, energy does not cycle, but rather it flows. Energy makes a one-way trip from the sun through these various aspects of the ecosystem, and then 100% of that energy eventually dissipates as heat. Heat is a form of energy, but it's a very disordered form of energy and not a form of energy that's easily used. So, if we look at this picture from our text, we see energy coming from the sun to plants, which we refer to as producers. Since they produce the chemical energy, they turn solar energy into chemical energy that can be used by animals. So during this process, some energy is lost as heat. In fact, some energy is lost as heat before the plants even take it up. A good portion of the energy that comes from the sun is simply reflected back up into the atmosphere. Next, what we see is this, that the energy that's taken up by the moose in this case is turned into mechanical energy in the form of kinetic energy, and a lot of energy in that process is lost as heat. So interestingly, what we see is that as this energy changes form from one form to another, in other words, as it's turned into chemical energy in the form of plants, and then as it's turned into mechanical energy and also some chemical energy in the form of the moose, then we lose a great deal of it. In ecology, we understand that approximately 90% of the energy is lost. So yeah, you heard that right. 90% of the energy is lost as we go from one form to another. We see that reflected in the second law of thermodynamics. The second law of thermodynamics is also known as the law of entropy. Entropy we could understand as disorder. And what the second law says is that when energy is changed from one form to another, there's a loss of energy, or at least there's a loss of usable energy. So we understand from the first law that we're really not losing energy overall, but we are using the energy that's available to be used by biological systems. Biological systems can only use ordered energy. And so energy that's stored in the form of chemical energy, glucose for instance, is a very highly ordered form of energy. Heat, on the other hand, is a very disordered form of energy that's not usable by biological systems. So, the second law of thermodynamics clearly states that as we go from one form to another, the disorder increases, hence the name law of entropy, and the amount of usable energy goes down as we go from one form to another. We see that illustrated on our slide. So we have carbohydrate, which is a very ordered form of energy. It's taken by the muscle, turned into ATP, which is then used by the muscle, which represents a transformation. And during that transformation, a great deal of the energy that was originally in the carbohydrate is lost in the form of heat. So we would expect approximately 90% of the energy to be lost in the transformation from the carbohydrate to the muscle. And of course, when we say lost, we don't really mean lost overall. We mean transformed into a more disordered form, a form that has more entropy and cannot be used. And we know that that form is heat. So we make our way now to a related topic, but we are shifting gears a little bit. We're going to see these previous topics that we talk about. We're going to see their importance in these topics, but we are shifting gears just a bit. Now, We introduced the term metabolism at the top of this slide, and so we use the term metabolism in biology to describe all of the chemical reactions that occur within the cell. And we understand that reactants are turned into products in these chemical reactions. And if we look at all of these reactions that are occurring, all the products that are being formed and otherwise, then we assign the term metabolism to describe the sum of all those chemical reactions. We then go on in the bottom bullet point here to make a distinction between the two types of reactions that occur in the cell. One type of reaction is an exergonic reaction. This is a reaction that gives off energy. The second type of reaction is an endergonic reaction. This is a reaction that requires energy. And so, interestingly, what we see in the second half of this definition is this. We are, of course, creating products in any reaction. And so in an exergonic reaction, because the reaction gives off energy, the products have less energy than the reactants. Products have less energy than the reactants in an exergonic reaction because energy has been lost in this reaction and given off in the form of heat. So to see that illustrated nicely, we go to the bottom portion of our slide, the nice picture we have from our text, and we see the energy referred to as Gibbs free energy, which is the energy that's available to use by biological systems. So the more energy you have there, the more energy is available to be used. Heat is, of course, not calculated in Gibbs free energy. And so what we see is that in an exergonic reaction, the reactants have quite a bit of energy, and the products have very relatively speaking, less energy. And so we have given off energy in this exergonic reaction. On the other hand, in an endergonic reaction, what we notice is this, that the reactants have a relatively low level of Gibbs free energy. And so we have to add energy to this reaction. And so the products have a much higher level of energy than did the reactants. And hence, this is an endergonic reaction. This is an reaction that requires energy. Now, interestingly, what we notice is that both endergonic and exergonic reactions are important in biological systems. And what the cell actually does is to pair exergonic reactions, reactions that give off energy, with endergonic reactions, reactions that require energy, in order to get work done. In fact, what we notice is that ATP, the molecule with which you are familiar, is a key component of these paired reactions, exergonic and endergonic reactions. Because as you realize, if we follow our little picture here on the slide, the transformation of ATP to ADP is, of course, an exergonic reaction. And so we have released energy by removing this third phosphate and transforming ATP to ADP. In doing so, and that's what this little side arrow indicates, we have made energy available to power endergonic reactions, such as protein synthesis, nerve conduction, muscle contraction, and actually anything else that occurs within the cells of the human body or any other living organism. So all the work that's done, in other words, in any living system is powered directly, at least, by the transformation of ATP to ADP, which is an exergonic reaction, and it provides energy to power endergonic reactions. Then the problem becomes, well, eventually we're, of course, going to run out of ATP. So somehow we have to get from down here to ADP back up to the top to ATP. So what we do then, of course, is to take energy from other exergonic reactions, and for instance, we'll provide a hint for what's coming up in our chapters in Chapter 8. Cellular respiration is a series of reactions, represents a series of reactions that are exergonic, and they provide energy to take us from ADP back up to ATP. And so this is essentially how the cell works. We are constantly breaking down food that comes into the body in the process of cellular respiration, and this is an exergonic reaction. That energy is used to take ADP to ATP, and then ATP is broken down into ADP, and that energy is used to drive all the reactions that occur within the cell. Now, we could ask the question, well, what the breakdown of ATP allows what to happen, and it's sort of a trick question. Everything that happens in the cell, and therefore in the body, happens as the result of the transformation of ATP to ADP. So, here are some examples that are provided by our text. Chemical work. So, for instance, the energy that's needed to synthesize macromolecules. Take hemoglobin as an example. So you are producing 2 million red blood cells per second. Each one of those red blood cells has over 1 million hemoglobin proteins. And all the energy that is required to synthesize each of those hemoglobin proteins is provided by ATP. Transport work being another example. We recently talked about the nervous system and the fact that active transport allows your nervous system to function. Think about that, the pumping of those sodium and potassium ions back and forth across those membranes of the nervous system. And so that's another excellent example of the role of ATP. In fact, there are many studies that indicate this may be the largest single expenditure of ATP that your body engages in, the active transport via the sodium-potassium pump of sodium and potassium ions back and forth across the membranes of all cells, but particularly the cells of the nervous system. And then finally, an easier one, a more obvious one, is mechanical work. And so think about the contraction of your muscles, as is being shown on this particular slide, or the beating of a flagella in a microscopic organism. So take a look at the muscle that we have here in this picture from our text. And so we have myosin here and actin up here. We've talked about actin before in our class. But your skeletal muscles and smooth muscle and cardiac muscle in slightly different forms are composed of myosin and actin. And so these little myosin heads that we see here, which look like small golf clubs, undergo these things called power strokes. And power strokes are very short movements. In other words, what's going to happen, as is portrayed in this picture, the golf club-shaped myosin head is going to be cocked. It's going to be pulled back into an active state via the expenditure of ATP. So we see that happening here if you follow the pointer on the screen. So ATP is expended, and then the little golf club-shaped myosin head is moved back a little bit, and then, so you can see here that ATP has become ADP and phosphate. Then myosin binds to actin and undergoes this little thing called a power stroke. And the power stroke is sort of like a mousetrap snapping shut. So the myosin head snaps back to its original shape after being pulled back, as you would pull back a mousetrap, by the expenditure of ATP. And this, and no other mechanism, is how your muscles contract. All your muscles, but especially skeletal muscle in this exact form. Smooth muscle, cardiac muscle, it's the same thing, but the arrangement is a little bit different. So the take-home message is, how many functions in your body involve ATP? And the answer is all of them. Anything that requires energy in your body requires the exergonic reaction of ATP being transformed into ADP to make that released energy available to be used by all these different functions that we see listed on this slide, and many, many more, by the way. We now come to an interesting topic in biology, and one of the most important topics that we do find in all of biology, the topic of enzymes. What we notice is this, that enzymes are typically proteins. There is a small exception that we'll talk about later in our class, in which RNA serves as an enzyme, but almost always enzymes are proteins. And furthermore, what we see is this, that the reactants of an enzymatically catalyzed reaction are referred to as substrates. And so all we need to do here is think, okay, I know what reactants are, and so when an enzyme is involved in a reaction, then instead of using the word reactant, we use the word substrates. Furthermore, we notice this, that enzymes speed up reactions, something that we've likely always known. And we also want to realize this, something we'll talk about more in some of the pictures that we have here in a moment, but each enzyme is specific. And so enzymes are not generic in any way, but each enzyme is required to speed up a particular reaction. And it's all the result of the shape of the enzyme. These are, of course, very, very small, but think about how a lock fits into a key. As we'll see in a moment, this is the analogy we use to describe how substrates and enzymes interact with one another. Furthermore, what we notice is this, that enzymes are typically arranged into these things that we refer to as metabolic pathways. Metabolic pathways are long chains of enzymes that eventually accomplish the production of a particular product. So what we see here is this. We have substrate A, which is transformed by enzyme number 1 into substrate B. Then substrate B is transformed by enzyme 2 into substrate C, and so forth all the way down the line until we get to G. So interestingly, you may ask, why do you need all these enzymes? And so the answer is, well, you don't always, but most of the time you do, because A is quite different than G. And we're trying to build a fairly complex molecule, so it takes a lot of different steps to get from A all the way down there to G. We want to take just a moment to look at an animation of how enzymes work as we build this image in our minds, this model, we might say, of how enzymes function. So this video is linked to the text in the upper right-hand side of the previous slide. And what we notice is this, that it gives us a nice little animation
During
of
the
enzymes
normal
and
enzyme
how they
catalytic
work.
cycle,
So on the
substrate
slide,
encounters
we
an
see the
enzyme.
enzyme and the substrate. And so the enzyme is shown in blue. Remember that enzymes are most typically proteins, almost always in fact. And here we have the substrate, which could be a protein, it could be or any of the other types of biological molecules with which you are familiar. Furthermore, what we notice is this, that on the enzyme itself, we have this region referred to as the active site that I'm outlining here. And so this explains the specific nature of enzymes, the reason that enzymes will only interact with a particular substrate. It's all about the fit, the overall shape here. So watch what's going to happen. So as we push play, we're going to see the substrate bind to the enzyme at the active site, and it fits. Like a key fitting into a lock, it fits. Like a puzzle piece, fitting in. When you finally find that piece that fits in a portion of a jigsaw puzzle, you feel great, and that's the kind of fit that we have here. It's very, very specific, and therefore it's very rare that an enzyme will catalyze multiple reactions.
The enzyme then facilitates the breakdown of the substrate.
There we have the products
which
being
apart
formed
from
and the products
enzyme,
leave.
leaving
Next
the active
substrate
site
comes,
free
it
to
binds,
catalyze
it's
another
turned
substrate
into
as the cycle begins.
next substrate comes, it binds, it's turned into products, and those products leave.
Again.
And so this happens over and over and over again. And the enzyme remains unchanged as a result of this interaction. In fact, our best estimates are that this reaction occurs somewhere between hundreds to thousands of times per second, if you can believe that. We now return to this idea of metabolic pathways in which enzymes are involved and build on this idea just a bit. So we've talked about the overall role of metabolic pathways and how each enzyme must be intact in order to get from A to G. There would be 1, 2, 3, 4, 5, 6 enzymes that are present in this case to get from A, the initial reactant, to G, the final product. However, what we want to add is this, that in reality, most metabolic pathways are highly branched. We have a very small, simple example that's shown in the lower left-hand side of this particular slide. This enzymatic pathway, though, I've chosen to use as an example because it really illustrates the importance of enzymes in our body and what happens if these enzymes are not formed correctly as a result of a mutation. Remember the central dogma of biology, DNA to RNA to protein. So a change in the DNA sequence results in a change in the protein enzyme. Now, in this particular case, we have the metabolic pathway that starts with phenylalanine, an amino acid, and ends with melanin. So melanin is the pigment that's found in all living organisms, including humans, that gives us the color that we see when we look in the mirror. And so each of us have a slightly different type of melanin, and that gives us the different phenotypes that you see when you look at different human beings. And then, of course, when you look across the natural world, the different colors of organisms that you see are a result of a plethora of different types of melanin. However, what we notice is this, that if one of these enzymes goes bad, then it has a profound impact on the overall metabolic pathway. Let's take a look at this picture on the lower left-hand side of our slide. In this case, we have two potential mutations and therefore defective enzymes that can occur. Enzyme number 1 can be mutated. I suppose we should say that any enzyme could be mutated, but the reality is that what we notice is that there are 2 enzymes of the 6 shown in this pathway that are commonly mutated. And what we see is that if enzyme number 1 is mutated, then you can't take phenylalanine and get to tyrosine, at least in the same concentration that you could previously. Now, there are other the pathway is somewhat redundant, and so there are other ways of getting to tyrosine, and so this doesn't mean that the individual will be totally without melanin, but what it does mean is that since you don't have as much enzyme going down the pathway that's illustrated by enzyme number 1 here, or that is catalyzed by enzyme number 1, you have more product that is following the pathway catalyzed by enzyme number 2, and you have this buildup of phenylpyruvic acid. In human beings, a mutation in enzyme number 1 is referred to as having fetal ketonuria, abbreviated PKU. If you look at the back of a diet drink can or a diet drink bottle, maybe more likely, like a Mountain Dew or a Diet Coke, you'll see that a little notice that says this product contains phenylalanine. And you may say, well, why does it matter if this contains phenylalanine? It matters for people that are fetal ketonurics, that have PKU. And so these people can be just fine if they regulate their diet quite closely and consume less phenylalanine than other people. If they don't do that, if they don't do that in childhood, if their parents don't do that for them, then something very scary takes place, and they develop with severe mental retardation. So now that we're aware of that in developed countries, this is one of the so-called inborn errors of metabolism that are checked for at birth. And, of course, if the baby has PKU, then the parents have to closely regulate the diet, then eventually the child will take over. But that's why you see some food items labeled as having high concentrations of phenylalanine. If you regulate your diet closely, there are no problems whatsoever. It's common enough in the human population that those labels are placed on food items. Interestingly, enzyme number 6 is less commonly mutated in humans, but we do see it in animal populations and occasionally in humans. And so if enzyme number 6 is completely mutated, then there's no production of melanin, and we have what's referred to as an albino organism. And so you're familiar with albino organisms in the pet world. So albino mice actually are very commonly bred so that they can be distinguished from wild mice. Albino snakes are common, and you're familiar with the phenotype. Albino rabbits, white with red eyes, is the phenotype that occurs when melanin is absent. So the bottom line is that both of those disorders, PKU and albinism, occur as a result of a mutated enzyme, a change in the DNA sequence that changes the protein and leaves the protein non-functional and unable to produce the products that it should produce. We go now to a more detailed discussion of enzymes and how enzymes work. And so what we see is this, that it's very difficult for molecules to react with one another at temperatures that you would find in living systems unless they're activated in some way. So this is an interesting idea, and we see this idea illustrated on the screen. So right now we're only talking about exergonic reactions, reactions in which the products have more energy than, the reactants have more energy than the products. And so overall, it's a downhill trip as far as energy goes. We start up here with the reactants, we end up down here with the products. But what we notice is that in these molecules, there's this hump here if we follow the cursor on the screen. This hump is referred to as the energy of activation. This is energy that's necessary to allow the reaction to occur. Once this energy is supplied, and you can see this hump here, then a great deal of energy will be released as a result of the reaction. So you go all the way back to where you started, and you go all the way down here, and this energy is released into the environment or to another molecule. And so we could think of it as we could go back to the ball on a hill analogy. If we had a ball up here, it wants to roll to the bottom of the hill, but it can't because of the hump that's in the way. Now, of course, if this was the opposite, if we had an endergonic reaction, if the energy of the reactants was down here and the energy of the products was up here, well, there's no chance the ball's going to roll up the hill, so we just wouldn't even have this discussion. But enzymes can be involved in allowing exergonic reactions to occur, reactions in which we have more energy in the reactants than we do in the products. And what enzymes do is the following. They lower the activation energy. So interestingly, it's as if, let's go back to the ball analogy on the hill. So we have this ball here, and we want to push it to the bottom of the hill. Well, so it'd be very difficult to push it over this blue hump here. which is the energy of activation in the absence of an enzyme. But when enzymes come along, they take this blue hill and they shift it all the way down to this yellow or kind of brownish hill that we have here. So it perhaps would be possible to push the ball up over this little hump and let it roll down to the bottom of the hill. And this is exactly what enzymes accomplish. Now, you may say, well, how would this possibly happen? But think about the diagram, think about the video, the animation we looked at a few minutes ago. And so what enzymes do is the following. They run into a substrate and they take that substrate and they do something, providing a microenvironment that allows the reaction to occur. So they might provide a little pocket that has a different pH. They might, and this is probably a bit more common, they might provide some pressure. They might twist the bonds of the substrate just a little bit so that those bonds break. It would be very unlikely those bonds would break if the substrate was not in contact with the enzyme. But as the substrate runs into the enzyme, it twists those bonds a little bit, causing those bonds to break. And so enzymes are essential for reactions to occur in living systems. Now, you could make the argument, well, what if you just heated things up? Could you supply the activation energy through the addition of heat? The answer is yes, but the problem becomes, as we'll see in just a moment, that living systems cannot tolerate an addition of heat, at least an addition of heat that would be sufficient to supply this activation energy. And that would result in destruction of the molecules that make up the system. So the only way in a living system that you're going to allow these reactions to occur is with the addition of enzymes. So we see this illustrated, what we just talked about on the previous slide, nicely here in this slide as well. Now, let's look at the bottom two pictures first. So the bottom two pictures show exactly what we saw on the previous screen. We have the reactant, which actually is called a substrate in an enzyme catalyzed reaction. We have the activation energy, and then we have, because it's an exergonic reaction, the energy that's released before you get to the products. And so we can see that with the addition of an enzyme over here, this is a non-catalyzed, this means no enzymes. We use the word catalyze to describe what an enzyme does to a reaction. It lowers the activation energy, making it more likely the reaction will occur. Over here, we have a catalyzed reaction, and we see that we've lowered the activation energy. And this means that up here, what we see is this, that we have a number of reactant molecules or substrate molecules on the y-axis, and we have this bell-shaped curve that we see here, and then we have the energy of the reactants found on the x-axis. And so when the activation energy is quite high, that means that a very small number, a very small percentage of the overall reactant molecules have enough energy. In other words, only the molecules that are found from this point, where I'm pointing with the cursor now, and up, have enough energy to undergo the reaction. But as we go over here to the right-hand side, what we see is this, that when we provide an enzyme, we take that activation energy and we slide it way down to the left. And because the activation energy is lower, we haven't changed anything about the molecules themselves, but we've simply lowered the standard. And so this means that a much higher percentage of the molecules that are present can now participate in the enzymatic reaction. So enzymes allow reactions to occur, and this is a key term, by lowering the activation energy. They lower the activation energy by providing a little microenvironment within the active site that allows the reaction to take place. All right, so moving on, what we see is this, that the interaction between an enzyme and a substrate is an interesting one, and it's a very intimate interaction that's very specific. So we see the substrate, we see the enzyme, and we see the transformation of the substrate into products here. And we see a bit of a better illustration, but remember, all these things are drawn very simply for you as beginning students to try to illustrate the interaction between these two things. In fact, the reaction is quite complex, and we do use this term lock and key fit. In other words, the shape is just more complex, that's all. It's not any more complex than what we're describing here. It's just a complex shape. And it's like a very complicated key that fits into a lock perfectly. And therefore, that makes enzymes, as we've said a number of times, specific to a particular reaction. But another thing we have to point out, and this is going to seem contradictory to what we have just stated, is this: that when the substrate binds to the enzyme, it changes the shape of the enzyme ever so slightly. We refer to this as induced fit. So I like to think of it as everything being made of Nerf, and you can imagine a substrate binding to a Nerf. I guess the substrate would have to be a little bit harder, maybe made of plastic, and it binds to this Nerf enzyme. And as it does so, it changes the shape of the enzyme ever so slightly, which therefore allows the substrate to fit perfectly within the enzyme. Now, it's still true that it's a very specific fit and that enzymes are very specific, but the induced fit model nonetheless plays a role in our understanding of how substrates and enzymes interact with each other. So it is a lock and key model, but the induced fit model does play a role in helping us understand how these two things interact with one another. All right. What we notice is this, that enzymes can both degrade and synthesize. So if we look at the top reaction from our text, we see that we have a substrate and we have an enzyme, and they bind together, and the substrate, which was one molecule, is transformed into two products, and everything leaves, and this happens again. So this, of course, is degradation. On the other hand, at the bottom portion of our slide, we have two substrates, one enzyme again, and the substrates bind, and one product leaves. And so enzymes are used for both tasks. They're used to break down molecules, and they're used to synthesize molecules within the cell. Finally, we get to one of the more exciting aspects of enzymes. And what we see is this, that this explains many things that we see in the natural world and within our body. So the rate of an enzymatic reaction is impacted heavily as a result of temperature. And we always see this bell-shaped curve that we see over here on the left. So on the x-axis, we have temperature, and on the y-axis, we have the reaction rate. And what we notice is that the reaction rate is very low at a low temperature, and then as the temperature increases, it goes up and up and up and up and up, and then it peaks. And then as the temperature continues to increase, it goes down and down and down and down and down, and eventually the reaction goes down to absolutely zero. And so we ask ourselves, why do we observe this particular pattern in the relationship between enzymes and temperature? And the answer is twofold. So the upslope that we see here, if you follow the pointer, is explained by this. There's no magical force, no attraction, magical or non-magical, I suppose, that draws enzymes and substrates together, but rather their interaction is the result of a random collision. Imagine substrates and enzymes, thousands of them, moving together in a single drop of liquid. Occasionally, a substrate runs into an enzyme, and when it does so in the correct, just in the right way, then it hits the active site and a reaction occurs. But the reaction doesn't occur unless that substrate hits the enzyme just so. And this means that at a very low temperature, all of the particles, including the substrates and the enzymes, are moving very slowly. And so as we start to increase the temperature by adding heat, we speed up the movement of both the enzymes and the substrates, and therefore, and this is a key term, increase the number of enzyme-substrate collisions. Enzyme active site collisions, I suppose, rather substrate active site collisions, I suppose we should say. So, we go up and up and up and up, and then eventually we peak. And so this is the point at which we have reached the maximum temperature at which the enzyme can withstand. So everything's moving quite rapidly at this point. And you could certainly ask the question, why doesn't this extend on forever? But what starts to happen, as you probably already surmised, is that the enzymes start to fall apart. And this is what we refer to as denaturation. So when we heat a protein up enough, then the enzymes fall apart. And now, as we continue to supply heat, a larger and larger percentage of the enzymes are falling apart, and therefore the reaction rate goes down and down and down and down and down, as we see illustrated on our screen. So this explains this bell-shaped curve that we observe in the interaction of temperature and the rate of enzymatic reactions. And it explains many things that we see in the natural world. For instance, look at our green iguana here. So green iguanas are awesome animals found throughout mainland Central, South, and South America. And they've invaded the southern portion of our country as well. Now, they are an exotic invasive species, and so we shouldn't be too excited about them. But I got to be honest, it's kind of hard not to be a little excited to drive down a street in Miami and see as many as 35 of these guys, which is the most I've ever counted, basking there on the sun just right beside the street in the middle of a city. These are big guys. They get to be 5 feet long or so. And so typically, they're really, really fast. If you've ever tried to catch an iguana, you've probably had an experience, at least these green iguanas, like I have. And so you walk towards them, and they're incredibly fast, and they just run and jump in the water, and I've never been able to get anywhere close to them. However, in certain rare instances in the middle of the winter, it will get a little bit cold down in South Florida. And when it does, you'll see these warnings that are posted by NOAA, which is the National Oceanic and Atmospheric Association, and they post falling iguana alerts. These iguanas like to climb trees, and so when it gets cold, their enzymes slow down so much. Their enzymes are pushed way to the left here that they can no longer hold on to the trees or really function at all. They're just basically asleep there, and they fall out of those trees. So it's very rare to find a reptile or an amphibian or any animal that's ectothermic, like we see on this slide there, that doesn't regulate their body temperature, that's found in cold environments. There are a few exceptions, but those are mostly areas in which you have a very brief warm period, such as maybe the Sierra Nevada mountains at high elevations in California there, where you do find a few species of amphibians. But mostly we find ectotherms in warm environments. On the other hand, the polar bear that we see here is an endotherm, and so it has no such problems as we just described there. It is constantly regulating its body temperature, and so it's holding itself right there where its enzymes are operating maximally, right there at the tip of the curve, not going too high and not going too low. We also notice this, that just like enzyme activity is affected by temperature, it's also affected by pH. And the explanation here is a little bit simpler, but what we notice is the following, that each enzyme has an optimal pH at which it works. Pepsin is found in the human stomach. Pepsin works best at a pH of 2 to 3, which is the pH that's found in the human stomach. And as you go below that and above that, then pepsin denatures. The protein falls apart and it can no longer function. Interestingly, also found in your digestive system, but in the small intestine, is trypsin. In the small intestine, we have a pH of 7 to 8, and so trypsin works great at this pH of 7 to 8, but it falls apart nearly completely when you get to the pH of the low 7s or 6, or a pH much above 9. And so here, there's no two-sided explanation like we had in the case of temperature. There's just one explanation. Proteins can only maintain their shape at a certain pH. As you leave that shape, then the protein falls apart. Lastly, we can impact the activity of an enzyme by changing the substrate concentration. So what we notice is that we can increase the frequency of an enzymatic reaction by increasing the substrate concentration because we create more enzyme substrate collisions, but only to a point. Eventually, we will saturate the enzyme. And so we go up and up and up and up, but then we level off because at that point, the enzyme's working absolutely as fast as it can. And so at that point, the only thing that we could do to speed up the reaction would be to provide additional enzyme. One thing we notice in the human body is that it's very important for cells to be able to regulate enzyme activity. And cells have multiple mechanisms, therefore, by which they can regulate enzyme activity. We go over a couple of these right now. So one possibility is that cells can regulate the presence or absence of an enzyme. In other words, the enzyme can be totally absent until it's needed, and then the DNA that produces the enzyme can be read, the protein produced, and so the enzyme shows up. We observe this in some enzymes within the cell. However, it's more common to see the second bullet point here in enzyme regulation. Cells can regulate the concentration of an enzyme. And so this means that an enzyme is always found within the cell, but at very low concentrations, and then when it's needed, a great deal more of the enzyme is produced. We observe this in enzymes such as detoxification enzymes that are found within the body. So, if you've ever eaten a burnt marshmallow, then what you have noticed, actually you probably haven't noticed it, but it did occur, is that your liver started to produce more of a particular enzyme referred to as cytochrome P451A in response to the consumption of those burnt products. So CYP1A, cytochrome P451A, the short version of that, CYP1A, is an enzyme that's highly inducible, and the body regulates its concentration greatly in response to need at any given times. Another possibility is this. The enzyme is always present, but it's activated or deactivated based on need at any given time. So one possibility for doing this is using cofactors. And so cofactors are typically organic molecules that are used by the enzyme in order to function. And so if they're absent, then that means that the enzyme is not able to function. So these molecules are required to activate the enzyme. This is actually the role of vitamins, as we'll build on the next slide as well. You may wonder, why can I eat a little vitamin and it has so much impact on my health? Well, it's because it's a cofactor that allows an enzyme, particular enzyme, to function. Lastly, for now, another possibility in regulating enzyme activity is phosphorylation. This is quite common. So what we notice is this, that phosphorylation occurs when another enzyme, interestingly, adds a phosphate group to a second enzyme, and by transferring that phosphate group that has been cleaved from ATP, it's transferring energy. That energy is used to, for a brief period of time, activate that enzyme and allow it to function, and then that enzyme will once again be deactivated, probably when the phosphate group is removed by a second enzyme that is there to deactivate that particular enzyme. So 3 enzymes total. One that's doing the job, one enzyme that activates it, and one enzyme that deactivates it. Now, here we see a nice little illustration of cofactors. And so this is a more grown-up diagram that's provided to us in our book by our text. And each of the little blue spheres, of course, represents an amino acid. And what we see is that the blue spheres represent the enzyme, the red spheres represent the cofactor, and so you'll notice the diminutive, the very small size, the cofactor relative to the overall size of the enzyme. And then the yellow spheres represent the substrate. And so in order for the enzyme to function, the enzyme is most of the physical mass of this entity, but the cofactor, which is much, much smaller, must be there in that active site. And this is an important thing as well. The cofactor is not used up by this process, not consumed by the process, but rather it functions as an entity necessary for this enzymatic reaction to occur. So a good illustration of this is scurvy. What we notice is this, that historically, sailors that went to sea for long periods of time would eventually become ill in some cases, depending on what they were eating. But they didn't understand that at the time. They simply noticed that sailors, like the gentleman in this drawing here, would eventually start to lose their teeth, their skin would become very pale, their eyes would sink into their head, and their gums would start to bleed, and then they would die. And they weren't really sure why they were dying. And to be honest, we didn't really come up with a reason, a mechanism for why this was happening, but we did figure out, and I mean the term we as human beings, so human beings did figure out that if we provided a very small amount of citrus fruits to each of these sailors, so just one every 2 weeks or so, that it would prevent this disease entirely. So what we now realize is that these sailors were suffering from a lack of vitamin C. Vitamin C is the cofactor in the reaction for the enzyme in the reaction that allows the production of collagen. Collagen is a very large structural protein that's found in almost every part of your body. And so it holds your skin together, it holds your teeth in place, it holds your bones together, it provides support for your blood vessels. And so when you're not able to make collagen and replenish the collagen supplies of your body, what you notice is that your body just starts to fall apart. And that's exactly what scurvy is and why these sailors were dying. And sailors eventually came to be referred to as limeys in England because limes were one of the things they had on hand and would keep very well in these barrels because of their very low pH. And so they found that by providing limes to the sailors periodically during these long voyages, they completely got rid of scurvy. And they got rid of scurvy because they were providing the cofactor for the reaction that leads to the production of collagen within the body. And so this illustrates the production of cofactors and furthermore illustrates the need for vitamins. Now, it is likely, nutritionists tell us, that living in a developed country as you do, that you are taking in certainly enough vitamin C, unless you're just only eating junk food. But that's the idea of vitamins. Whether they're necessary or not, a multivitamin wouldn't hurt anything, at least, in making certain that you have all of these various cofactors found within your diet. As we progress in our discussion of enzymes, we're going to talk for just a minute about reversible enzyme inhibition. Talking about two types of reversible enzyme inhibition, furthermore, competitive inhibition and non-competitive inhibition. To do this, we're going to look at our animation that was linked earlier in this lecture and that we looked at to understand the interactions between enzymes and substrates. So here we see that in the latter part of this animation, we see an illustration of competitive and non-competitive inhibition. We begin with competitive inhibition. So what we see is we have an enzyme and we have our competitive blocker here. So this is not the substrate, but rather this competitive blocker is going to bind to the active site. Watch this. There it goes. Now the actual substrate comes along, and when it tries to bind,
the substrate is encountered, the active site
it bounces right off, so there's no reaction. And so this is competitive inhibition. The more of the competitive inhibitor you have, the less reaction that you have taking place. So this might be a little confusing looking at it in terms of this one little diagram here. You have to imagine, if you can, hundreds or thousands of these enzymes and hundreds or thousands of these blockers and actual substrates. Therefore, the speed at which the reaction will take place is all about the relative concentration of the blockers versus the real substrate. So if you have more real substrate, then the reaction is going to proceed at some speed. But as you increase the concentration of these blockers, which are non-competitive inhibitors, which are competitive inhibitors, then the speed of the reaction is going to go down and down and down, as you could imagine. However, an important thing to note is that competitive inhibition doesn't completely stop the reaction. On the other hand, we also have non-competitive
and
inhibition.
No reaction will occur.
So there we have another one bouncing off in competitive inhibition. So this is non-competitive inhibition. And so what happens in this case is we have an enzyme that binds to another site on the enzyme referred to as the allosteric site.
Away from the active
and so the allosteric site is down here. It's not the active site. But when that non-competitive inhibitor binds, look at that, it changes the shape of the active site.
Causes a
and this means
change in the enzyme,
that
altering the shape
when
of the active
the
site,
enzyme
which
comes
the
along and
substrate
tries
from
to bind, the substrate
no
comes
reaction
along
will
and tries
occur
to
as
bind.
long as
It's
the
unable to
non-competitive
bind,
blocker
but
is
it
bound to
bounces
the
right
enzyme.
off, as we see there. So, non-competitive inhibition has the potential to shut down an enzymatic reaction entirely, depending on how much is present. So once the non-competitive inhibitor binds, as long as it's present in that allosteric site, then the reaction is not going to take place. Lastly, what we see is this, that allosteric inhibition can be used as to accomplish feedback inhibition, which is a form of negative feedback. So if we look at the diagram on our slide, on the right-hand side, what we see is that we have the original substrate and the enzyme. And so substrate A binds to the enzyme, and substrate A is turned into B and C and D and E and so forth until finally you get the end product F there. The end product F is going to go around and bind to the allosteric site of the enzyme, just like we saw in that video, and it's going to change the shape of the active site, preventing the substrate from binding, the original substrate A, from binding to the active site. So it's a nifty way of regulating the amount of product that an enzyme will produce, or a series of enzymes in this case. As long as you have enough F is going to shut down the production of this metabolic pathway by binding to enzyme number 1 and preventing A from binding. Now, when F gets used up or degrades or goes away, you're going to need more of it. And so that works out perfectly because F is no longer no longer going to be there to bind to the allosteric site. And so, although it's not shown in our diagram here, you're going to have a change in shape of the active site back to its original form, and substrate A will be able to bind to it, and the whole metabolic pathway will resume production of F. Now, there are molecules that bind irreversibly to enzymes. These molecules tend to be quite dangerous because they bind and they don't let go. Everything we've talked about so far, whether it's a competitive or non-competitive inhibitor, it binds and then it releases. So molecules like cyanide bind to enzymes and then they don't let go. And so cyanide is extremely dangerous because it binds to enzymes that are involved in the production of ATP that we'll talk about in Chapter 8. And that's why cyanide leads to death in a very short period of time as a result of the fact the body cannot produce ATP. And as you know, you only have enough ATP at any given time to last about 60 seconds or so. Nerve gas is another scary example. And so interestingly, many types of nerve gas, such as sarin, nerve gas being, if you're not familiar with it, a horrific weapon that we as humans discovered back in the early 1900s, and it has a long and horrific history, used some in World War I and in later times as well. But nerve gas can be, as you have probably seen illustrated in movies, placed in a rocket, shot, it explodes and kills everyone in the vicinity. And so it works because it shuts down acetylcholinesterase. Acetylcholinesterase is an essential enzyme in the human nervous system and every organism's nervous system, by the way, because it breaks down acetylcholine and allows the relaxation of muscles. If you don't have it, then you have the sustained contraction of muscles. They never relax, and that eventually leads to death of the organism. So, molecules that are irreversible inhibitors tend to be quite dangerous because they bind and shut down enzymes permanently. Now, at the very end of our chapter, in the last few pages, we kind of provide a teaser for the next two chapters, Chapter 7, Photosynthesis, and Chapter 8, Respiration. And what we notice is this: those chapters deal with oxidation-reduction reactions. The story of especially respiration, but also photosynthesis, is the story of passing electrons, moving electrons around from one molecule to another. And as we do that, we see that we have particular terms we use to describe that process. The molecule that loses an electron is oxidized. The molecule that gains an electron, as you know, is reduced as a result of the fact that the gaining of an electron results in the addition of a negative charge and an overall reduction in the charge of the molecule. Therefore, the molecule is reduced. Both of these obviously have to take place at the same time. One molecule must give up an electron for another to receive it. And what we notice, as I mentioned, is that the story of photosynthesis and respiration is the story of the moving of electrons from one place to another. In the case of photosynthesis, we remove electrons from water, add them to carbon dioxide to make glucose, and as we remove the electrons from water, excitingly, oxygen is produced. That's just really an accident of photosynthesis. And so plants really don't need very much oxygen. They need some as a result of the fact that they do have mitochondria. But because that's part of photosynthesis, then the atmosphere in which we live is about 21% oxygen today, meaning that it's suitable for animal life to survive. Next, we see cellular respiration. And in cellular respiration, interestingly, we're going to remove electrons from glucose, and we're going to attach them to oxygen. And so as we remove them from glucose, glucose falls apart, creating carbon dioxide. As we add the electrons to oxygen, we create water. And so it's not essential that we understand the significance of the movement of these electrons at this point, but the movement of these electrons is going to represent the transfer of large amounts of energy. In fact, it's this energy that your body uses to make ATP and that is sustaining life in your cells at this particular moment. Lastly, what we see is this. Excitingly, in these discussions about photosynthesis and respiration, we're going to get to talk about ETCs, electron transport chains. And essentially, what we're going to see is this, that an electron transport chain is a series of carriers. It's like a series of steps that allows us to gradually release energy from electrons and harness that energy for use. So in the process of respiration, we're going to have some really high energy electrons, and we're going to take those electrons and we're going to take them from this step, if you follow the pointer, to this step, to this step, to this step, and then finally to this step, which is the attachment of these electrons to oxygen. And at each step, we have lost a little bit of energy. We could go from point A, if you're following the pointer, the high energy electrons, down to point B, the low energy electrons. But if we did, there would be this big flash of energy, and it would be very difficult for the cell to harness that energy. By using an electron transport chain to go in a step by step by step manner, that allows us to harness the energy that is removed from these electrons to make ATP. In fact, we take that energy and use it, if we follow the pointer here, to establish an electrochemical gradient. In other words, as we remove energy from the electrons, we use that energy to pump an item. This item in particular is a hydrogen ion, from one side of a membrane to the other, creating this reservoir of hydrogen ions on one side of the membrane. We then allow the hydrogen ions to flow back down their concentration gradient, allow them to diffuse, a term that you know. And this process is referred to as chemiosmosis. So it's called chemiosmosis because as they go down this concentration gradient, we take ADP, adenosine diphosphate, and make ATP. In a similar manner to water, how water flows through a dam and turns these big wheels that we call turbines to generate electricity. Well, the hydrogen ions flow down this concentration gradient, and the energy that they release is used to add that third phosphate back onto ADP, creating ATP. This is a topic that we'll discuss in much greater detail, excitingly, in Chapter 7 and especially in Chapter 8. Here, in the last slide of our lecture, we see the process of chemiosmosis illustrated. So here, we have our low hydrogen ion concentration, and we're pumping. This is one of the carriers of the electron transport chain. As the energy is removed from electrons, we use that energy to pump the hydrogen ions over here to this high hydrogen ion concentration environment. The hydrogen ions flow back through this cluster of proteins referred to as an ATP synthase complex. And as those hydrogen ions go through, we use the energy that they are releasing to take ADP, add this one additional phosphate, and make ATP. So the energy in your body, the ATP that's formed in your body, is formed as a result of this process of chemiosmosis.